Hi everyone, this is the pBLU gene cloning lab. In this lab, you're going to do gene cloning, which is a really cool, useful method used everywhere in the world in biotechnology and scientific labs. And in this experiment, you're basically going to change the traits of bacteria. You're going to work with E. coli, and you're going to get the chance, if you were actually doing the lab, not with the quarantine, you're going to put DNA or a plasmid specifically into E. coli that has an antibiotic resistance gene. And so you're going to make bacteria antibiotic resistant. So we're going to talk about all of this. And the plasmid is called pBLU, the DNA that you're inserting into the E. coli. This is why it's called the pBLU lab. And to start out with a little bit of background, gene cloning is a biotechnology method that we use in science to get bacteria or yeast or any mass producer, an organism that can reproduce a lot, to make us something useful. So you take a gene of interest that you want to make, such as insulin. Let's say you want to produce insulin, or you want to produce lactase, the lactase gene, to give to people who are lactose intolerant. You're going to isolate it from an organism, such as a human. If we're talking about insulin or lactase, you're going to transfer it to a mass producer, meaning an organism that can produce a lot of copies of itself, like bacteria, because they divide. Specifically, E. coli divides every 20 minutes, and you're going to get it to produce whatever it is you want to produce, such as insulin or lactase or whatever you want to make. And it's going to make a lot of copies of that, and that's what gene cloning is. So you can get mass producers, which are typically bacteria, to make many things for you. That's what gene cloning is. And with gene cloning, we produce a recombinant organism, meaning an organism with a combination of DNA. So a recombinant organism is any organism with DNA from two or more sources. So if you take a cow and you put chicken DNA in it, that becomes a recombinant organism. If you take bacteria and you put another human DNA in it, like a human DNA piece or fragment, that's a recombinant organism. And we're going to talk about this example with making insulin in a second. So as you can imagine, gene cloning is very useful, and it's done to you create new traits in organisms. We use it every day to produce medication. So insulin is a medication that we give to individuals with diabetes, produce different vaccines, produce commercial proteins, produce organisms or fruit and vegetables that are bigger, riper, tastier, last longer, are resistant to different pests, weeds, all of that. So gene cloning is very useful, and you can get your organisms to glow. You can insert a gene that makes organisms glow, and a lot of times people do this to study infection. So if you have bacteria and you want to study where it's infecting, you could insert a gene that gives it the ability to glow, and you could trace it. So an example of gene cloning is producing insulin. So we take the human insulin gene, we isolate it, it's pretty easy to do. And then you take a bacteria, you take out its DNA, its plasmid DNA, and you put your human insulin gene into the plasmid. So remember, with the biotechnology, we said that you cut both DNA pieces with restriction enzymes, which are enzymes that cut. Then you get them to ligate together, so the human DNA gets put into the bacterial plasmid DNA. Then you take that plasmid that you just made, called the recombinant plasmid, because it has two different sources of DNA, and you put it back into your mass producer, which here is bacteria. So now your human gene is in the bacterial plasmid that's put back in the bacteria, and the bacteria reproduce, making a lot of copies of insulin. So they make the insulin gene, which gets transcribed and translated into the insulin protein. Then you isolate that insulin protein and you create human insulin to give to diabetic patients and other patients that may need insulin. So this is how gene cloning is useful. You're going to do a form of gene cloning in the lab. We're going to talk about it. So gene transfer is done by transformation. To actually get your DNA back into, so in this step right here, when you make your recombinant plasmid, to get it into your organism, you have to transform your organism. That's what transformation is. This is what you're going to do in the lab. Transformation is transferring DNA into a competent cell. A competent cell is a cell that's able to take up DNA. So generally, a cell cannot just take up DNA. It has to have a membrane that is somewhat permeable so that it can take up DNA. That's what transformation is. There's natural transformation and artificial transformation. Some bacteria have the ability to do natural transformation, meaning without you doing anything to them, bacteria naturally can take up DNA from the environment. So an example of this is H. pylori bacteria. They can, if they're in the environment, they can just take up pieces of DNA. They're able to do that. In lab, we're going to do artificial transformation. Whenever we say artificial transformation, that's when you in lab can take organisms and give them the ability to be competent or their cell membranes to be permeable so that they can take up whatever DNA you want them to take, such as insulin, such as lactase, such as a gene that's going to make a strawberry bigger and redder. So that's what artificial transformation is. It's making recipient cells competent or their cell membranes a little bit permeable, because you have to make the cell membranes permeable so they can take up the new DNA. And you can do artificial transformation by multiple ways. We're going to talk about two ways. You can heat shock the cells in the presence of positive ions. So the cell membranes are generally negatively charged. If you take an organism and you add some positive ions and you heat shock them, meaning that you take them and you put them heat ice, that's what heat shocking is. You make the membrane a little bit permeable so that they can take up the DNA that you pipetted on them. So if you have bacterial cells in a tube and then I pipette or add DNA that I want them to take up, such as the insulin gene plasmid or another plasmid I want them to take up, I have to do something for them to be able to take up that new DNA or new plasmid. So you can heat shock the cells with chemicals that are positively charged. Another way you can make the cells competent is by electrocuting them. We say electroporation using high voltage. Now, both of these, whether you're heat shocking the cells or you're doing electroporation, they have to be very quick. You can easily kill the cells if you overdo it. I've definitely done it as a grad student with both. So with both heat shocking and electroporating, you're very careful. You're timing yourself. It's 1 to 2 minutes, depending on what you're doing, but not longer than that. If you do either method for longer, you're going to kill the cells. You just want to make the cells a little bit permeable. You don't want to kill them. So that's transformation, is getting your plasmid in. So if we have our bacteria and we add our plasmid of interest that has antibiotic resistance, that has insulin, that has the ability to cause the organism to glow, not grow, you're going to get it in by transformation. And again, you can do that artificially by heat shocking the cells or electroporating them. So with artificial transformation, you're using a plasmid of interest that you're trying to get in your bacteria. And your plasmid of interest has the gene that you want. So it could have a human insulin gene. Again, it could have a glow gene. It could have whatever. From the title of this lab, you're going to use a plasmid called pBLU. And as you can guess from the name, it caught with some things that I'm going to talk about later, it causes the bacteria colonies to look blue. That's why it's called the pBLU plasmid. And we use plasmids because they can self-replicate. They have something called an origin of replication. So if we focus on a plasmid, if you look at this here, this is our bacteria. We're trying to get a plasmid in. This plasmid could hypothetically be this plasmid. And they're often high copy. You can buy different plasmids. Usually we use plasmids that are high copy number plasmids, so they can make a lot of copies of themselves in the recipient, which is bacteria. In this lab, you're going to use a plasmid called pBLU. It's a genetically engineered plasmid, meaning it's a plasmid that a scientist or a group of scientists have made. So when we look at the pBLU plasmid, which is a very popular plasmid used in gene cloning, it's popular because it has a few things that makes it relatively easy to use. So scientists, when we want to do experiments, we want to do the simplest, easiest thing possible that works. So pBLU is a good plasmid. It has an origin of replication, the ORI, and this is important because this lets the plasmid self-replicate. Because when you get the plasmid in the bacteria through artificial transformation, you want it to make copies of itself. The plasmid also has an antibiotic resistance gene. We call it a marker gene, and it specifically has the antibiotic resistance to ampicillin. So it has the ampicillin resistance gene, meaning when you put this plasmid in bacteria, it will be resistant to ampicillin. Ampicillin should kill bacteria, so it should kill E. coli, but when you give them this plasmid, it won't kill them anymore because now you gave them the ability to break ampicillin. It also has a gene that codes for beta-galactosidase. It ends in ASE, so you know it's an enzyme. Beta-galactosidase, or the LAC-Z gene, that's what it is. It's an enzyme that breaks down the sugar lactose into glucose and galactose. So lactose is a disaccharide, and LAC-Z, or the beta-galactosidase gene, can break down lactose so that bacteria can use it. This is another marker gene, and it's going to become important in a second when I talk about why we have that gene on there. And then you also, on every plasmid, there is an area that has a lot of restriction sites where you can cut open the plasmid. So I can cut open the pBLU plasmid and, why is this jumping? Okay, I can cut the pBLU plasmid and add an insulin gene here if I want to make it. I could cut it open and insert the flagella gene, for example, to make bacteria motile. I can do a lot of things, and this is called the restriction site area. It's called the MCS in science, multiple cloning site. So there's a lot of areas here that allow you to insert foreign genes. So this is what our pBLU plasmid has on it. It has 4 things. It has an origin of replication, it has ampicillin resistance gene, it has the beta-galactosidase gene, or the LAC-Z gene, and it has restriction sites, an area for restriction sites, which is also called the MCS. If you look at the picture of the plasmid from your lab manual, here where you see like, so for example, here there is, Clor. These are names of different restriction enzymes that are used that you can buy so that you can insert your gene. This is too much detail for you guys, but I'm going to say, if you want to insert your gene, you're not going to insert it in the middle of your beta-galactosidase gene or the ampicillin resistance gene, because you'll break those genes. So in this plasmid, this area right here is a good area to insert my gene because it's not going to affect the ampicillin resistance gene, and it's not going to affect the beta-galactase or LAC-Z gene. So the goal of this lab is for you guys to take E. coli cells and to insert pBLU plasmid into them. That's your goal. And you're going to do this by artificial transformation. And once you get your pBLU plasmid in E. coli, your E. coli are considered transformed cells, and they're cells that now are resistant to the antibiotic ampicillin, and they also have LAC-Z. So E. coli, generally the bacteria is sensitive to ampicillin, meaning that it dies when you give it ampicillin, and it doesn't have beta-galactosidase. So you're going to make recombinant E. coli with the pBLU plasmid. You're giving E. coli new genes. You're giving them ampicillin resistance, and you're giving them the beta-galactosidase enzyme. So to get the plasmid into E. coli, you have to artificially transform them. What we're going to do in lab, we're not doing electroporation. You need these machines to do electroporation. You're going to heat shock your cells in the presence of calcium chloride. So you're going to add calcium chloride to your cells, and you're going to have your E. coli cells and your plasmid together in a tube. You're going to add some calcium chloride, then you're going to heat shock them. And I have a picture of that we can talk about. So with heat shocking, maybe I do not have a picture actually. Anyway, once you have them together, you're going to heat shock them. So you have your tube, you have your E. coli, your plasmid, and your calcium chloride. Now I want to actually get the plasmid in the E. coli in that little test tube. So you're going to heat shock it. So you're going to take your tube and you're going to go from heat to ice to heat really quickly. You're doing each for like 1 to 2 minutes, a few seconds, and that's it. You're done. Hopefully your E. coli cells have taken in your pBLU plasmid. Now, by looking at your test tube, you have no idea if your transformation worked. That's why we say transformation is never 100% efficient. To determine if your E. coli cells actually took on the plasmid or were transformed, that's what that means, you're going to have to see, do they have the plasmid characteristics? And remember, when we look at our pBLU plasmid, it gives the cells ampicillin resistance and it gives them the beta-galactosidase gene. To assess if your plasmid actually went into E. coli, if transformation worked, you're going to grow bacteria on medium with ampicillin. You grow your bacteria on agar. So now we're going to have agar plates that have ampicillin added to them. You're going to grow your transformed cells. Your transformed cells should be able to grow on that agar plus ampicillin, because if they did take up pBLU plasmid, it has ampicillin resistance gene. You're also going to see, did they actually take up beta-galactoside? Do they have beta-galactosidase? Did the transformation work? How you're going to test this is, remember, beta-galactosidase is an enzyme that breaks down lactose. We can add an analog of lactose, meaning something similar to lactose, in our media, and the analog is called xgal. You're going to have your agar, it's going to have xgal on it, and then you're going to plate your transformed cells. If your transformed cells were actually transformed and they have the beta-galactosidase gene can break xgal, and when you break xgal, you get a blue color. So if bacteria are blue, they have the beta-galactosidase gene. So the procedure, you're going to follow the lab procedure. You're going to take to make it simple, you have your test tube. You're going to add a little bit of E. coli cells. You're going to add your plasmid, and then you're going to add calcium chloride. You're going to heat shock the cells, meaning you're going to take your tube that has everything and go from ice to heat bath to ice. And then now you're going to hope that they actually got transformed. Then you're going to add some broth so they can recover. And then you're going to plate them on different agar plates to see, did your cells actually grow and did they actually get transformed? Did they take in the pBLU plasmid? So you're supposed to grow your transformed bacteria and your control bacteria on different plates, and you're supposed to make predictions. So you have a bacteria that has no pBLU in it, a tube of that, and a tube that does have pBLU. So when we look at the bacteria that does not have the pBLU plasmid, just E. coli. So forget pBLU, just E. coli. When we plate E. coli on regular agar, we should see growth. If we plate E. coli on agar plus ampicillin, we should not see any growth because ampicillin will kill the E. coli. If we plate E. coli on a plate that has ampicillin and xgal, again, we should see zero growth because this plate has ampicillin and E. coli cannot grow on ampicillin. So again, to recap, if we take just regular E. coli, not the transformed one, nothing to do with pBLU, and you plate it on a regular agar plate, you're going to see growth. If you add ampicillin to that agar, you should not see any growth because ampicillin is an antibiotic and it kills bacteria. And if you plate it on a plate that has ampicillin and xgal, which is just a lactose analog, you should again see no growth because of the ampicillin. Now, if you take your transformed E. coli, your E. coli that has the pBLU plasmid, and you grow it on different plates, this is what we should see. So remember, the pBLU plasmid has ampicillin resistance and beta-galactosidase gene. So if you take your transformed E. coli and you grow it on a regular agar plate, you will see growth and you'll just see our regular white colonies. If you take your transformed E. coli and you grow it on agar with ampicillin, you should see growth if you actually did transform your cells, because remember that the pBLU plasmid gives E. coli ampicillin resistance, and you'll still see just regular white, yellow colonies, whatever you guys call them. And then if you take the transformed or recombinant E. coli, the E. coli with the pBLU plasmid, and you plate it on a plate that has agar, ampicillin, and xgal, you will see growth because your pBLU plasmid gave the bacteria the ability to be resistant to ampicillin, but you'll also see blue colonies because the plasmid has the beta-galactosidase gene, and beta-galactosidase can break xgal and get a blue color. So that's the theory behind it. Those should be your results if transformation actually worked. And as a side note, this is really hard to do. Transformation is a pain. Sometimes cells take up your plasmid, sometimes they don't. So if I were working for a biotech company and I come and this plate had all white colonies, which happens most of the time because things are not perfect, you have to redo the experiment because for some reason or another, your bacteria did not take up the plasmid. They just grew because you still have E. coli. So the objectives of this lab is you guys should finish this lab knowing what genes are on the engineered pBLU plasmid. So it has an origin of replication, it has ampicillin resistance, it has beta-galactosidase gene, and then it has a multiple cloning site or restriction sites. Explain how you introduce the plasmid into the host E. coli. To get your plasmid, your pBLU plasmid into E. coli for this lab, you're doing artificial transformation and you're specifically doing, you're heat shocking the cells with calcium chloride. So you heat shock the cells. Remember, you're trying to get the pBLU plasmid in, and the calcium ions are positive, so they neutralize the negative charges on the plasma membrane. And then interpret gene expression on the plates. So when you look at plates, what do blue colonies mean? What do white colonies mean? What does it mean that just E. coli, why did E. coli not grow on the agar plate with ampicillin? It doesn't because it's not resistant to ampicillin. Why did the recombinant E. coli with pBLU grow on ampicillin agar? Because it does have the resistance gene. And what is a recombinant organism? So remember, it's an organism that has more than one source, two sources of DNA. So a source of DNA from another organism, or two organisms, or three. And what are methods of artificial transformation? You can get plasmids into bacteria by electroporating them, by heat shocking them. So just have an idea of how this experiment works. I really wish we were doing this experiment in lab because this is one of my favorite experiments because it's so relevant to the biotech research world. And that's it. That's the end of this lab. So here's the pBLU plasmid, and I hope this makes somewhat of sense, even though you guys are not doing it. And I'll end it here.